Hello students, welcome to my another online session. I am Mr. Avinash Kale, Department of Microbiology, S.V.K.T College, Devali Camp. Students, we will learn in today's lecture about the additional aspects of immunity. This topic belongs to your paper number 2 of TYBSc Microbiology, that is Immunology. In this topic, we are going to learn the further aspects of innate immunity. In previous lecture, we have learned the basic thing about the immunity. In immunity, we have seen that it is one of the important mechanism of the every vertebrate organism to protect its body against the attacking by foreign infections. In immunity, we have seen the different type of the classes of immunity. In that classes, we have seen the different types and working of innate and acquired immunity. This innate and acquired immunity are two principal types of immunity, and in that we have seen how they work at different level to fight against the different types of pathogens. Now, in today's session, we are going to learn that humoral component of innate immunity. We have seen the innate immunity is a natural immunity where the person has got it in the form of gift from Mother Nature, where the different type of the physical, chemical and other cellular factors are present, where these components are working for actually different purposes, but they use the additional effect, additional advantage to that individual by protecting that part or whole the body against the attack by the infections. So in this innate immunity, we have seen how it works at the different type of the level. In previous sessions, we have seen that this innate immunity works at the physical and the mechanical type of the barriers, which is commonly called as a first line of defense mechanism. But in today's session, we will see about its internal type of the parts, where the humoral components, which are soluble in nature, which are flowing in the fluid portion of the body and giving the protection to the each corner to that body from the infection of that or infection caused by the organism. And these components that we will focus mainly as a pattern recognition proteins, which are famously called as a PRPS, and pathogen associated molecular patterns, that is PAMPs, or they are also called as a PAMPs. Now let us see in detail each of these humoral component, their location, their working, where they are secreted, and actual mechanism, how they destroy that pathogen in details. Now, in innate immunity already I told you that it is a natural and non-specific immune response against the foreign bodies. It works at physical, mechanical, physiological as well as at the cellular level. It provides the first as well as second line of defense in that individual against the infection. Then, however, despite of this, there are various other soluble and insoluble components present in the fluid portion of the blood, which are also responsible for giving the innate protection to that individual against the different type of the infection. So these humoral components are mainly defensins. Already I told you, pathogen associated molecular patterns or PAMPs. These are the molecular structures of the microorganisms which are pathogenic in nature, which are required for the survival. Means these are produced by that pathogenic organism. We are calling them as antigenic in nature, which are harmful to our body. These are present in the molecular form, and these are produced by them, and they are recognized by our innate immunity as harmful, and against them the protection is given to them here. Then another is a damage associated molecular pattern or the DAMs. So these damps are actually produced during the course of inflammation or tissue injury or different type of the cell damages caused outside or inside the body. And these damaged parts itself are acting as a stimulator to work that type of the immunity. That's why they are called as a damage associated molecular patterns or the DAMPs. Then others are called as a cellular receptors. These cellular receptors are present on each cell of our body. In immunology or in immunity, all the cells are mainly grouped into two categories. That is, immunological cells, means their main role is to fight against that infection by reacting with that antigen or by catching that antigen and destroy them. That's why they are called as a immunocytes. And other cells, means other than the immunocytes, they are called as the body cells. They are the non-immunocytic cells are there. That is our common tissue cells are there here. So these all cells produces the receptors. The receptor are the specific type of the, you can say, the templates or the molecular type of the structures are there here, who are recognize the specific type of the molecules present on the microbial cell or the antigens, and they get stimulated by them. And after stimulation, they initiate such kind of the physiological, chemical or molecular mechanism so that the destruction of that antigen or that foreign agent is carried out. These cellular receptors are commonly called as a pattern recognition receptors. So they are pattern recognition receptors or the PRRS, means that specific antigen or that pathogenic microorganism secretes the molecule or possesses the molecule on their surface, and there is a specific type of the molecular pattern on them. And against that molecular pattern, our body, cells of our body possesses the receptor on that here. And the interactions are taking place once that pathogen has entered inside the body. And when the interactions takes place, these interactions are the intermolecular interactions are there. These interactions itself send the signal to the nucleus of the cell or the cytoplasm of the cell, and from that signal the different type of the destructive immunological mechanisms or molecules are produced which stop the further multiplication or infection of that pathogenic microorganisms. So these are the PRRS or the cellular receptors. Other type of the component, humoral component, is a complement. These complements are the proteins present in the blood of the every type of the individuals here, which comes in the action after stimulation by that antigen, and by the sequential action finally they destroy the type of the antigen or they initiate the other secondary type of the immunological responses, especially the cell mediated or the antibody mediated type of the responses here. Another type of the mechanism is the kinins, which are the secretions of the cells. Then other mechanism is a acute phase reactants. So these all components are called as a humoral components because they are soluble in the fluid portion of the blood, because humor in Latin means the liquid or the fluid portion. So they are flowing in that fluid portion, and due to that here there is a production is taking place. So let us see about the pathogen associated molecular patterns in detail in this session, which are commonly called as a PAMPs. This pathogen associated name itself indicate these are not produced by the body of that individual. They are produced only by the microorganisms, especially disease causing microorganisms. So these structures are usually fundamentals to the integrity, survival and pathogenicity of the microorganisms. Means these structures are basically produced as a weapons in the terms of clinical or medical microbiology. They are called as a virulence factors. They are produced as a virulence factor by that pathogens, and these are shared by the entire classes of that pathogens. They are present on the surface or they are produced in in the secreted form by that pathogens to protect themselves against the immunity or the different type of the antimicrobial type of the agents here. And these PAMs are expressed constitutively in the host, and they detect the pathogen regardless of their life cycle of the stage. Means these PAMs, when they are produced by the pathogen, so in the host where they are entered, so they are detected by the immunocytic cell and non-immunocytic cell, okay, at any stage of that type of the pathogen, and against them there is the initiation of different type of the innate immune responses, or they may activate the cells of the acquired type of the immunity to destroy them by carrying out the cytotoxicity. So this type of the PAMs are the germline encoded, means they are encoded during their embryonic stage, and their production is continuously going on throughout the life of the type of the individual here. They are non-clonal. Non-clonal means here they are not expressed in the form of group. They remain localized to that specific type of the cell, and they are expressed on all cells of that specific type of the given type. If the one specific cell, for example, the connective tissue cells is expressing that here, so after the expression here, all the connective tissue cells are producing this type of the PAMs, and are responsible to destroy that type of the pathogen here. These PAMs don't have any kind of the immunological memory. Means when they are produced, so they are not stored in the memory of that immunological cells here. So when that same pathogen re-enter against them, again this mechanism is again reinitiated here. That's why they are not stored in the immunological memory here. So they are the important features of the PAMs here. Now in this slide you will see this is a electron micrograph of E. coli, which is a gram-negative bacterium, and this is a molecular design of cell wall and cell membrane of this E. coli structure. So E. coli is also one of the pathogenic microorganism, and in the PAMs you will see on the surface of this E. coli these structures which are floating, the chains which are floating here. So this is a lipopolysaccharide, which is a chemical virulence factor produced by this E. coli organism, which is called as a endotoxin. Remaining part is a structural proteins which are present in the embedded form in the cell wall and cytoplasmic membrane of this E. coli. Now in this another structure, this is a structure of the slide of the electron micrograph of a Staphylococcus aureus, which is a cocci shaped bacteria, and these cocci are present in a bunches. And this bacterium is also one of the pathogenic microorganism, and it is a gram positive in nature. So in the gram positive bacterium here you will find again there is a presence of this virulence factor in this chemical form. Okay, they are the glycolipids, then the glycoproteins are there here, and the peptidoglycan is there here. So these structures which are present on their cell surface, this pattern itself are acting as a stimulator, which are responsible for the activation of the different type of the cells of the immunity. Due to that here their destruction or their further multiplication cannot take place. So whenever this organism enters inside, so this chemical structure, what are the patterns which are present on them in their molecular form, so against their pattern there is a presence of the specific type of the receptor sites on the immunological as well as non-immunological cells of the body, and they itself send the signal to that cell for the activation and their destruction by such type of the manner. Now they are the different type of the pattern recognition proteins and damage associated molecular patterns. If you see that these chemicals are mainly nucleic acids, proteins, cell wall lipids, carbohydrates, or straight-induced protein, crystals, nuclear proteins. So these are commonly present in the form of their cellular structures like single-stranded, double-stranded RNA, pillin, flagellin, which are the components of pili and the flagella, lipopolysaccharides, lipoteichoic acid, mannan and glycans, like this way here. And this kind of the molecular patterns, which are actually their chemical structures, but they are acting as an antigenic in nature or toxic in nature here, and against them cells of the body in their humoral part there is a presence of the particular immunological type of the mechanism to destroy them here. Means this PAMPs and DAMPs itself are acting as a molecular signal when that pathogen enters inside the body. That pathogen may be the virus, either bacteria or fungi or mix of this here. When they enter, so whatever the chemical structure they brought with themselves, which is got to them by Mother Nature, so they itself are acting as a signal to activate these cells of the different type of the cells of innate as well as the acquired immunity. So these cells are get activated and they further initiate the mechanism to destroy them or to stop their further multiplication. Now let us see the other type of the particular humoral component, that is a pattern recognition receptors, which are called as a PRRS. So chemically they are the several families of protein. Chemically they are proteinic in nature, but depending upon their chemical structure, their organization, their conformation here, it is a family of the several different type of the proteins. These proteins, these PRS, are located on many effector cells of our immune system, most importantly on the professional antigen presenting cell. This antigen presenting cell name itself indicates they catch the antigen which has entered inside the body, process that antigen in such a form they enhances the antigenicity of that antigen and present it to the effector cell like the T lymphocyte, B lymphocytes or other type of the cytotoxic type of the effector cells. Due to that, effective destruction, powerful destruction of that antigen is taking place. And majorly macrophages, dendritic cells and B cells are acting as a antigen presenting cell. So these PRS are generally located onto the professional antigen presenting cells and other type of the effector cells also. These pattern recognition receptors are generally present in the cell membrane of this cell, where they are embedded in that, where the some part is present in the cytoplasm and some part is floating on the surface. So they are generally called as a transmembrane pattern recognition receptors. And other kind of receptors are not present in the cell membrane, but they are present in the cytoplasm or cytosol. That's why they are called as a cytosolic type of the pattern recognition receptors. There are the three classes of these pattern recognition receptors. First is called as a secreted PRRS, means they are secreted outside the cell, produced inside the cell and secreted outside the cell. Next is called as a endocytic PRS. They are present inside the cell and they act inside the cell. And third is called as a signaling PRS, means when they are produced, they itself send a molecular signal to the effector cell. Due to that, the further effector mechanism to carry out the cytotoxicity are get initiated and the destruction of that antigen is taking place. Now let us see about the functions of this pattern recognition protein. Following are the principal functions of this pattern recognition receptors. First is called as a opsonization. So the opsonization is a very important mechanism in the phagocytosis, where on the surface of the phagocytic cell, these opsonins are present here. These opsonins are such a secretions or molecular secretions are present here, and against them on the surface of the phagocytic cell there is a presence of the receptors. So when antigen enters inside the cell, their chemical nature itself, which is soluble in nature here, against that chemical nature here, the phagocytic cell has a receptor, and the phagocytic cell stops there and get activated for the further steps of the phagocytosis. That process is called as a opsonization. Second, the pattern recognition receptor activate the different type of the components of complement. Complement is also one of the humoral component of our innate immunity, and it acts in a series, in a cascade like manner here. So this pattern recognition receptor itself are activator of the different type of the pathways of complement for destruction of that pathogen. Then phagocytosis means they help for the coming in action, immediate action of the phagocytosis. So they also help for the phagocytosis. Then they are also helping for the activation of pro-inflammatory signaling pathways. So in inflammation, different type of the pathways are involved, and to stimulate the inflammation, these PRS are also acting as a stimulator by giving the molecular signal to initiate the process of the inflammation process. And PRS also induce one of the important mechanism in our body, that is called as a apoptosis, that is a self-destruction of that antigenic cell or the abnormal cell of the body like the cancerous cell. So due to that here, the no more further harmful action of the cell continued here. So they are the functions of this pattern recognition proteins here. So let us see the each type of the particular pattern recognition protein. The first is the secreted pattern recognition receptors. So these are the examples of the pattern recognition receptors. So the secreted are commonly includes, they are the antimicrobial peptides. Commonly they are the alpha and the beta defensins or cathelicidins. They are produced or they are recognized on the microbial membranes which are negatively charged, and their role is to carry out the opsonization, microbial cell lysis, and they are acting as a immunological cells chemoattractant. Then the dermicidin, then regal gamma or reg 3 gamma is there here, collectins, then the minus binding lectins are there, then the surfactant proteins A and D are there, pentraxins are there, and C reactive proteins are there here. So these are recognized on the surface of this different type of the microbial cell membranes, and in the third column there it has the different type of the functions here. Now in this slide you will see that this is an antimicrobial peptide which is secreted by the type of the pathogen. Now when it is entered, so when it directly come in the contact with the particular that phagocytic cell, for example, or other immunocytic cell, for example here, so it has a weak type of the interactions here. But when they come in the contact with this type of the bacterial cytoplasmic membrane, so they come in the contact with this type of the cell and their opsonization is get enhanced. So by the opsonization here, what happen here, the different type of the mechanisms are get involved here, and due to that here the destruction of that microbial cell is taking place. In this another type of the mechanism here, this chemical peptide itself are acting as a chemoattractant by the electrostatic and hydrophobic type of interaction here. So they attract these different cytotoxic or effector cells of our acquired immunity, where like the cells like neutrophils, T cells, monocytes, mast cells, dendritic cells are get attracted there here. And when they get attracted here, so the microorganism who have carried these antimicrobial peptides on it, so these cells are get activated, and these cells further secretes their different type of the granular content on that microbial cell. So this is the surface of the microbial cell, and the destruction of that microbial cell is taking place, what we call it as a cell lysis. Now in this another slide, the other example is the collectins. which are commonly called as a mannose binding lectin or the MBL. So in this mannose binding lectins, these are the specific type of the chemicals which are secreted by the microorganism who contain the mannose as a such type of the chemicals in them, or on the microbial cell surface there is a presence of the these polysaccharides which are the polymannose or the mannose sugar which is present here. So against them, this is a C1Q. Okay, this C1Q is a one type of the particular component of the complement, and this component of the complement which is a proteinic in nature, which is a polymeric in nature here, it has such kind of the complex type of the structure, and on the tip of this complement component there is a presence of binding site. This binding site is responsible to bind to the IgG and IgM type of the antibody. That's why it is called as a IgG and IgM binding site. And this is a mannose binding protein. This mannose binding protein has the such type of the structure which are having the such type of the arms on it here. And this mannose binding protein, you see in this another picture here, this is a C1Q which is upper one like upper one here. So to that here, this binding, this is a microbial cell surface is present here at this downward here, and here the IgM antibody is bounded to that here, and this dark brown color are the microbial surface antigen which contain the chemical mannose or mannan, they are present in them here. So this molecular binding is taking place in between this C1Q component and this IgG or IgM type of the antibody here, and this itself is acting as a trigger to stimulate this complement component, and this complement component further initiate classical and alternative pathway and further carry out the destruction of that microbial cell surface by producing the membrane attack complex. In another picture of this type here is a mannose binding lectins are present here. So this mannose binding lectin, which is commonly called as a MASP1, MASP2 here. So this mannose on the surface of that microorganism is present, this brown color and yellow color here. So this mannose binding lectins which are present on this C1Q type of the components here, so they specifically recognize it and the binding is taking place here. And after binding here, it further initiate the complement cascade by alternative or by the classical type of the pathway, mainly by the classical pathway, and they produce the membrane attack stage, or they may act as a opsonins to activate the phagocytic cell, and then they carry out the destruction of this harmful type of the microorganism. So this is the role of collectins, which is a other type of the pattern recognition protein, which is a secretory in nature. Next is a pentraxin, which is also called as a C-reactive proteins. This pentraxins has a such type of the activity. So this is a pathogenic microbial cell surface. So this is secreted, this type of the CRP pentraxin on the surface here. This is the B phase and this is the A phase of this pentraxin here, and this is a cell surface of that immunological cell. On that here, these pentraxin receptors are present here. So they specifically bind to it here, and this binding, this interaction itself is triggering the process of the phagocytosis mechanism in that phagocytic type of the cell. Or in this other picture here, these pentraxins, which are C-reactive protein which are secreted here, they also has the binding site to the C1Q type of the first component of the complement in the classical type of the pathway. So that this binding itself is responsible for the activation of the complement, means when they are expressed and when this binding takes place here, so itself activate the complement, and this complement further initiate the classical type of the pathway, and further it either activate the phagocytic cell or it may produce the membrane attack complex here, and the destruction of that pathogenic cell is taking place. So this Pentraxins are playing a very important role in this type of the mechanism here. Second type of the pattern recognition receptors are called as a endocytic pattern recognition receptors, which are present inside the cell. Their example is a mannose receptors, whose code number is a CD206. Then the C-type lectin receptor. Then these are recognized as a carbohydrate. They recognize the carbohydrate is a mannose type of the carbohydrate they recognize. Especially they are present onto the bacteria, fungi and other type of the parasite on their surface, cell surface here. And this type of receptor against this pathogen is expressed onto the macrophages, and they are helping for the activation of the phagocytosis. Then the activation of the other type of the inflammatory component for their activation, they are helpful in nature here. It is like this way here. So this is a macrophage, one type of the phagocytic cell here, and on this here these receptors are present, you see, like this way here. So this is a LPS, that is a lipopolysaccharide receptor, that is a CD14 receptor is there here, is a T lymphocyte receptor. There are the different type of receptors are present here. And on this here, what happened, this endocytic, that is this receptor are produced on the surface, and they are responsible for the activation of the macrophages, and further activation of this macrophage leads to the enhancement of the phagocytosis. Now let us see about the another kind of the PRRS, that is a scavenger receptors. So they recognize the lipoproteins on gram-positive as well as gram-negative bacteria. The role of this scavenger receptor is to eliminate old as well as apoptotic type of the cells. The cells which are wrongly formed or which are cancerous in nature or virally or parasitically infected cells are there here. So they are eliminated by these receptors. They are mainly produced on the macrophages for the phagocytic purpose. So the role is for the phagocytosis and for the NF-kB activation. So just like in the previous slide, so they are expressed on the surface of the macrophages here, and these scavenger receptors are just expressed or shown in this diagram accordingly here. Now another kind of the receptor is called as a signaling receptor. The signaling receptor are the 4 major classes. The first is called as a toll-like receptors or TLRs. This receptor is a transmembrane receptor, means some part is present in the cytoplasmic membrane and some part is present in hanging position outside the cytoplasmic membrane. Then the C-type lectin receptor or the CLRS is there. Another kind of the transmembrane receptor is there here. And other is called as a NOD-like receptor or NLR receptor. These are present in the cytoplasm. That's why they are called as a cytosolic receptors. Fourth class is called as a retinoid acid inducible gene, that is a RIG-like receptor, which is also present in the cytoplasm. That's why it is another kind of the cytosolic receptor. Now this is a location of this different type of the receptors. So this is a toll-like receptor, this is a CLR, this is a endocytic receptor. So this CLR and TLR are the transmembrane receptor, this is a RLR. So these are the cytosolic type of the receptors are there here. Now let us see this one receptor in detail, that is a toll-like receptor or the TLR. This is a computer generated 3-dimensional structure of toll-like receptor. This green color and blue color, such a anchor-like structures you will find, they are the receptors. This white and red color are it is a cell membrane. Already I mentioned in the previous slide, this is a transmembrane receptor. It is a proteinic in nature. So this in Germany 1980, its first discovery was made in the Drosophila fruit fly embryo. The German meaning, this toll is a German word, its meaning is weird. So it is actually involved in the regulation of embryonic development of the Drosophila fruit fly. it has a cytoplasmic domain of 12 protein. It is homologous chemically and functional point of view. It is homologous to the interleukin 1 in the vertebrate type of the organism. Let us see this chemical structure. So this chemically it contains the transmembrane domain, that is the LLR, and the TIR domain. So in this diagram you will see this red color, which is a coiled structure, is there, which is the exterior domain is there, and this yellow color is a TIR domain is there here, where these middle structures are called as a cell membrane of that cell of that individual here, and the other is a ribbon model of this exterior domain is depicted in this diagram. Now let us see where this TLR or toll-like receptors are located in the cell. So they are located as an extracellular portion, where they are transmembrane, they are present here, where you can see they are interacting with the bacterial cell wall lipids, or they are interacting with the fungal polysaccharide, that is a lectin, or in cytoplasm. In cytoplasm, or they are called as a cytosolic type of the TLR, they are called as a NLR, PR, RLR, CDS, like this way. Means whatever the microbial cell structures or chemicals, just like a bacterial peptidoglycan, viral RNA, microbial DNA, so their interaction is taking place with them here. Or the endosomal type of the particular TLR are there here, where in the endosomes of the phagocytic cells, whatever the digested part or microbial cell parts, just like the DNA, RNA, or other structures are there here. So with them, these endosomal TLR are interacting with them here. Let us see the different types of this TLR. So they are expressed as a localized ligands here. So it is a full family as per their chemical composition, their localization, their function. As per that, is a full family is there here. So they are commonly TL1, TLR1, 2, 3, 4, up to the TLR13 is there here. Among these TLR1 to 9, they are expressed in the human as well as on the mouse cell surface. TLR10 is expressed onto the human cell surface, while TLR 11, 12 and 13, they are expressed onto the mouse cell surface. So they especially recognize the different type of the ligands of microorganism, like the triacyl lipoproteins of microorganism, lipoproteins, zymozyme, mannans, flagellin, lipoticoic acid, zymozyme, viral or bacterial, single-stranded or double-stranded RNA, they recognize here. Where after recognition here, they send the signals to that cell so that the digestion or inactivation of that whole microbial cell or their component they can carry out here. Now this is a location of a different type of this TLR here. So in the plasma membrane, TLR 1, 2, 6, 4, 5, 11, you can see how they are embedded. And in endosomes or in lysosomes membrane here, TLR 3, 7, 8, 9, 13 are present in the embedded type of the form. And in each, it is shown that what type of the particular molecular pattern they recognize. For example, TLR4 recognize the gram-negative bacterial cell wall components. TLR2 and 1 recognize the bacterian parasites, or TLR3 recognize the viral double-stranded DNA, and so on, that you can understand from this diagram by such type of the manner here. Now all this is about these different type of the humoral components of the innate immunity, which are working at the different type of the level for helping for the activation of this different type of the effector cells and other immunological mechanisms of the body, so that effective destruction of the pathogen can be takes place. So students, in this session we have learned the different type of the humoral components of the innate immunity, which are working at the different type of the level. So what we have seen in today's session, that there are other mechanisms other than the physical anatomical barriers which are working in the innate immunity. These mechanisms are commonly includes is a pathogen associated molecular patterns, that is PAMs, or pattern recognition protein, that is PRP or PRRS. And in that pattern recognition protein, we have seen in detail about the toll-like receptors, where they are located, what is their function, what are their different type of the classes. In next session, we will study the cellular components of the innate immunity. How different cells of immune system are working for the working of the innate immunity. We'll see in our next session. Thank you.